Welcome to the session 4 for the NPTEL course Cell Biology Cellular Organization, Division and Processes. This is basically a discussion session where we discuss the concepts that we've learned and discuss a previous year assignment question. I'm Smriti, a PMRF TA for this course, and I hold such discussion sessions every Monday 6 to 8 p.m. So today, this week, which is week 4 of the NPTEL course, basically dealt with mitosis. Mitosis is this process of cell division wherein there is equal chromosome division between the daughter cells. We have been learning about the cell cycle and its regulation for the past few weeks, and it culminates in mitosis, which ultimately results in the formation of two daughter cells. And this is the reason why the cell has gone through so much effort and replicated its DNA and ensured there is proper genetic material transfer to the daughter cell and so on. So as we've seen previously, cyclin are proteins that are translated in a cyclic manner. That is, there are different cyclins that come up in different stages of the cell cycle. And this is what ensures that there is a specific activity that happens in specific events that occur in different stages. We've seen that there is first a G1 stage when the cell is getting ready to replicate its DNA, and then there is an S phase wherein the DNA is replicated, the G2 phase where the cell is growing in size and getting ready for cell division, and finally the M phase, which is the mitotic phase, which we are going to see in this discussion session. Herein, the cyclin that is important is cyclin B in higher eukaryotic cells. And this is known as M cyclin as well, because mitotic cyclin, which forms the complex with the CDK1. Now in yeast, there is just one CDK that binds to different cyclin-like proteins. But in higher eukaryotes, there are different cyclins in different stages of the cell cycle. And in mitosis, CDK1 plays a major role. So once the M cyclin is synthesized, it forms a complex with CDK1. However, at this stage, there are phosphatase transferring proteins such as V1, which transfers phosphate groups to the like an inhibitory region, and other proteins such as CAK, which is CDK activating kinase, which transfers phosphate to the activating region. However, as long as this inhibitory phosphate transferred by V1 is present on this MCDK complex, this is inactive. So even though there is translation, the complex is not formed and mitosis does not progress. But in the presence of CDC25, which is a phosphatase, what it does is it removes this inhibitory phosphate, as we can see here, and the protein becomes the active MCDK complex. Now, how this works is there is a positive feedback wherein the increased presence of active MCDK further increases the activation of CDC25, and this generates more and more active MCDK, and there is progression into the mitotic phase. So one of the major things that the cell needs to ensure upon mitosis is equal separation of the chromatin of genetic material amongst the daughter cells. But as we know, in the interphase, the genetic material is present as this coiled structure known as chromatin, which is a complex between DNA and proteins, and it is tightly wound, and this is present as a messy, tangled structure of DNA. If in this state, the chromatin is pulled into two ends, as in what happens during mitosis, then there is high chances that this tangling may cause strand breakage. So in order to avoid this, the cell takes many measures, and to understand that, we need to know what chromatin is and how it was initially observed. Here we can see that if you look at the DNA within the cell under a microscope, we can see a beads on string structure. This is basically proteins associated with DNA that helps in its compact packaging. So this was first reported by this scientist known as Kornberg in the early 1900s. And herein he observed that when you take chromatin out from the cell and you digest them with nucleases, then you get a 200 base pair uh, strand on the gel electrophoresis. However, when you remove all proteins attached to this and you take the naked DNA and then you digest it with nucleases and run it on the gel, you get a smear. Now, what he basically did was a type of a nucleus protection assay in which what happens is, supposing this is your DNA fragment and there is some protein bound to it. Now, when you treat it with a nuclease, here they've used the micrococcal nuclease. If you use that, then only in the region that the protein is bound, there will be no cutting. Otherwise, the nucleus will be able to cut the DNA at every other region, because of which, if the DNA has no protein bound to it, there are very small fragments of differing sizes produced. This will lead to the smear. But if there is a presence of protein that binds to the DNA, then that region is not cut and there is a higher fragment of DNA that is found that gives rise to these gaps wherein we can see that there is some protein present. This is how he first reported that there was something known as a nucleosome, wherein what essentially happens is that the naked DNA is bound by these proteins known as histones, wherein 146 base pairs of DNA is wrapped 1.65 times around a histone core. This histone core consists of 8 proteins, 2 each of H2A, H2B, H3, and H4. And in the connecting region between two histone cores, there is histone 1 molecule that basically acts like a linker histone. So this is what he named as the nucleosome core particle. And this is what was visible initially as these beads that are seen on the DNA strand. Hence, we can the structure of a chromatin is basically, like I mentioned, DNA with proteins, and here it has DNA wrapped around the histone core and held in place by a H1 linker histone. So, however, upon mitosis, the cell puts in a lot of energy in order to condense this chromatin-like structure into these well-organized chromosomes, which we have all heard of and are familiar with. Now, in order to condense the chromatin as well, there are high chances that there are supercoils present or there is tangling present, and there are two very crucial means that the cell takes up in order to avoid this. One is the presence of topoisomerase enzyme, which we have seen in the previous class, wherein its major function is to release the supercoils that are generated. So say there is a supercoil over here, then the topoisomerase will be able to create breakages in the DNA fragment and release these supercoils, wherein once you cut, then both the ends can rotate independently of each other, and herein this supercoil nature is removed. Another means that it ensures to prevent tangling is creating domains in DNA. So what this means is, say there is a huge amount of long length of DNA present in our cell. Here it decondenses it one by one, as in if there are chromatins with histones present, the histones are released from a very short fragment of DNA only. This is replicated and then it is again condensed into chromatin, and then the next domain opens up. And so forth. The entire DNA is replicated and the entire DNA is present as two different strands, and they are not mixed together to form tangling. So there are two proteins, cohescence and condensins, that are very important in this two process. Now, we'll first address the question as to why cohesence and condensins are important. One is that you need equal partition of genetic material between two cells. If there is gene 1 in mother cell, in one daughter cell, there should be gene 1 in the other daughter cell as well. Similarly, every gene, one copy has to be present in this cell and one copy has to be present in this cell. Now the cell has ensured that it is replicating the DNA. Now if it completely loses the sister chromatids, then what happens is one copy will float off to one end of the cell, the other copy will float off to the other end of the cell, and equal partitioning will become impossible. So in order to avoid this, what the cell does is it has these proteins known as cohesins. They are made up of multiple proteins, as basically like a complex, which forms a ring-like structure. Now, this ring-like structure holds the two sister chromatids together. Sister chromatids, meaning the two copy of the genetic material encoding the same gene, is present right next to each other and is bound by this ring-like structure to prevent the floating away of the sister chromatids. The next question is, how is this chromatin, which is present with nucleosomes and all, how is it condensed to form this chromosome, which is even further compaction of the genetic material? In this process, the other protein that is important is condensin. Condensin, if you can notice, is very similar to cohesin and also forms this giant ring-like structure. But in this case, what it does is it forms the ring-like structure within the same sister chromatid, but with regions that are far away from each other. So say there is one condensin, it forms a ring-like structure and pulls other regions that are farther away. And ultimately, more and more condensins join in together, and we can appreciate the fact that once DNA that was loosely packed with the accumulation of condensin keeps getting compact, and ultimately leads to the formation of a chromosome. Now, for this, again, energy is required, and it has an ATPase domain over here, which is able to hydrolyze ATP, and that energy that is released is used to compact the chromatids. Now, we'll go through every stage of mitosis one by one with these microscopic images, which are very beautiful. Here, what we should appreciate is that these are the microtubules, and in red here is what is the chromosome. So in the prophase, if we can see, there is an intact nuclear envelope, which is not shown here, as in it's not stained here, but we can see that all the chromosomes that are present nicely within this boundary like region, which is the nuclear envelope. So the nuclear envelope is still intact. There is some formation of microtubule organizing centers. There's something known as a centrosome from which the microtubules are being formed, and then the chromosomes are present within the nucleus. They are condensed. They are not existing as chromatids anymore. Next phase is the pro-meta phase, and here is where the nuclear envelope actually starts breaking down. So what was initially like a boundary within which all the chromosomes were present, herein we can see that this boundary is lost and the chromosomes are just present all over the cell at this place. But here in this stage, there is a very clear formation of the centrosome, and the microtubules are already organized as the mitotic spindle. Now, this actually in this image we cannot appreciate, but the microtubules are already attached to the chromosomes. Next is the metaphase. Here we can clearly see the formation of the mitotic spindle. It's very clear. Additionally, there is organization of the chromosomes along the center of the cell, and the centrosomes are very distinct. The next phase is the anaphase, wherein the chromosomes are pulled towards the two corners, and this is the centrosome, and the microtubules keep shortening in length, because of which the chromosomes are pulled towards the end, and there is equal partitioning of the genetic material between the two daughter cells. And in telophase, the separation is complete. They have reached the spindle poles, and there are few microtubules still present between these chromosomes. One is the interpolar microtubules, but the set of daughter chromosomes are already present at the spindle pole, and slowly the cell will start entering into cytokinesis, which is basically where the cell divides its cytoplasm as well into two equal parts if it is a symmetric mitotic division, and the nuclear envelope starts reforming. So again, we can see that there is some boundary over here within which the chromosomes are present in two different cells at this point. And there is still a small joining between the cells, which when upon completion of mitosis will break, and it will become two individual cells. So these processes are just carried out by very very crucial regulatory events, and we'll just look into a few of that. So like I previously mentioned, MCDK complex is what initiates most of the processes that is needed for mitosis to progress. And as we saw in this microscopic images, the ones that initiate mitosis more like, or the steps that is very crucial to the initiation of mitosis, is one, formation of the centrosome at two ends, two, dissociation of the nuclear membrane. And this is what initiates mitosis. And MCDK complex is very important for this process as well, wherein what it does is it phosphorylates various components of the centrosome, and what was initially present in one place of the cell now matures and moves to two ends of the cells, forming the spindle poles. Then it phosphorylates the nuclear pore complex, many proteins in the nuclear pore complex, and slowly they disassemble. Then there is the structural unit within the nuclear membrane known as the nuclear lamina, which is also heavily phosphorylated, because of which it breaks down. And slowly in the pro-metaphase, we can see that there is no nuclear envelope that is remaining. So how this does is that, like I mentioned, first there is an intact nuclear membrane, and then slowly the nuclear pores are first disintegrated. And once nuclear pores are disintegrated, lamins are phosphorylated. And then upon that, the cell's complete nuclear envelope is broken down. But still they are present distributed along the cytoplasm, which later upon completion of mitosis can again fuse together and reform the nuclear envelope. So this is very important, because up until the nuclear envelope is non-broken, if you notice that the centrosomes actually do form microtubules, but the microtubules cannot reach the chromosomes. And unless and until it does not reach the chromosomes, it cannot attach to it, and neither can it separate. So the second major event in mitosis is the attachment of microtubules to chromosomes. So herein, we'll first see why these microtubules are necessary, and what basically is something known as biorientation, which is also very essential. So if this is the chromosome, we know that there are multiple parts that are characterized by the presence of special proteins that are concentrated in these regions. These are known as the arms of the chromosomes, and if you see this, these are sister chromatids that are associated together with each other, and they need to be separated into two different cells. Now, this end of the chromosomes are the telomeres, which we in the previous session discussed as to why it was necessary for solving the end replication problem. But in this mitotic event, what is important is the center region known as the centromere. The centromere are characterized by the presence of multiple proteins that are concentrated in this region to form this unit known as the kinetochore. The kinetochore is what attaches these microtubules to the chromosomes. Now, microtubules at one end are attached to the centrosomes and one end are attached to the chromosomes, which we will see more in detail. But this attachment is what ensures that the chromosomes are moved towards the poles. But still, what is important to notice is that one end of the chromosome needs to be attached to one pole, and the other end of this chromosome needs to be attached to the other pole, so that the same sister chromatid does not move towards one cell. It needs to split and move towards two different daughter cells. That is why biorientation is very important, and we will see why. So how this chromosome attaches to the microtubule is, I mentioned that there's something known as the kinetochore, in which one of the important protein is this NDC80 complex. This NDC80 complex on one end associates with the centromeric region of the chromosome, and on the other end is capable of interacting with the growing end of the microtubule. But what is important here is it does not directly interact with the end of the microtubule. It has this lateral orientation, which means that if this we can say is the end of the microtubule, it attaches somewhere in the inside end of it. This is important because microtubule keeps growing and keeps shrinking and keeps growing. that dynamic nature is very important to its existence. And so if it directly interacts with the end, then once when the microtubule is shrinking, then NDC80 complex will fall out. So in order to prevent this, it has a lateral orientation. And we can see that first, like I mentioned, in the prophase, when nuclear envelope is still intact, the microtubules cannot interact with the chromosomes. So from one spindle pole to another spindle pole, there are interactions. But as soon as the nuclear envelope breaks down, the microtubule grows towards the chromosomes and manages to attach to the kinetochore region by the help of this NDC80 protein. Now, again, one end is attached to one pole and the other end is attached to the other pole, because of which there is equal partitioning of the sister chromatids. This is known as biorientation. Now, if this biorientation does not happen, then the cell ensures that this interaction is not stable only. So say only one end of the spindle pole is attached to a chromosome, interaction is not stable. Say if both ends are attached to the same spindle pole, again, it's not stable. Say both two different ends are attached to the same sister chromatid, again, it is unstable. Only when there is proper attachment of one pole to one kind of and one pole to the other, there is a stable interaction. This, they have identified that this factor that decides if it's stable or unstable is tension. How this happens is, say there is a sister chromatid that is not properly oriented and not properly attached to the microtubule. Now, because of this, what happens is there is another component of the kinetochore. Like I mentioned, it's made up of multiple proteins, and one other protein is this aurora B kinase. And that is able to interact with the microtubule and phosphorylate it and release the NDC80 complex, because of which the interaction between chromosomes and microtubule is lost. However, if there is enough tension, that means that it is tautly pulled to both ends, then the microtubule is present nowhere near the aurora B kinase, and this is not being able to phosphorylate these proteins, and hence this chromosome is still attached to the microtubule itself. This is how the cell ensures that there is proper biorientation and equal partitioning of the sister chromatids. Another thing that is essential for the cell to ensure is to generate this something that is pulling the chromosomes towards the end. If we can see in this image, the chromosomes are not being floated to the ends, but rather there is some pulling force that is able to pull the chromosomes towards the end. Now, what is it that generates this force was of great interest to the scientists. And before we get into that, we'll just look at the different microtubules that are present in the cell so that we can understand how this experiment was designed in the first place. So if this is an entire cell, there are two spindle poles comprising of a centrosome. The centrosome is known as the microtubule nucleating center, and here there are a lot of microtubules that is emanated out of this centrosome. Some of these microtubules will directly go and interact with the microtubules generated from the other centrosome. These are known as interpolar microtubules. There are other microtubules that are generated but do not really interact with anything. These are known as astral microtubules. And finally, there are those that interact with the kinetochore, and they are the kinetochore microtubules. Herein, what happens is towards the centrosome, there is a minus end of the microtubule, and anywhere outside, there is a plus end of the microtubule. So near the chromosomes, there are plus end. In the end of astral microtubules, there's a plus end, and in between where these two interact, there is a plus end. Now, this is very important because this is what generates this bipolar nature and determines the polarity of the cell, and this is very important to ensure proper mitotic division. So there was a researcher who was interested in understanding a different aspect of the cell, wherein he was working on these axons, which is known as the squid giant axon, because it is a very long cell. But what the cell does is it's able to accurately move different proteins from one end to the other. There are also such cells that exist in the human body, like very long neuronic cells that can transport signals from one end to another in a matter of no time. Similarly, in this animal known as a squid, there's this giant axon that you can actually see with your naked eye in itself. And here, there is a cargo that is moving over long distances very quickly. So he was trying to understand this, wherein what he used this technique that we use in research known as in vitro reconstitution, which is basically we are trying to isolate certain compounds that is essential, mix it together in a test tube outside the cellular system and see if the same reaction happens. So say you want to see if one protein is generating ATP. You take the protein, you put ADP and see if ATP is being generated in an in vitro system in the presence of nothing else. So only when every component that is essential to a process is present will that reaction occur. And this is what he was trying to do, wherein he was trying to reconstitute proteins and other factors that are important to the movement of the cargo. In this process, he identified that microtubules is what is along which the cargo is moving, and hence he designed an experiment, the first experiment he designed, wherein he took microtubules, purified them. They were considered to be the tracks for the movement. Then, of course, you need energy to transport anything across the cell. So he added ATP, and finally the cargo. Now, if only these three were important, as in if only cargo and microtubule interaction was necessary for movement, he would have seen movement within the test tube, but he did not see any movement. So he concluded that there is something that is missing here. So in the next experiment that he set up, what he did was he added microtubules, ATP cargo, and along with this, he added the fraction of the cell which contains all the proteins present in the cell. So in this experimental setup, he saw that there was movement. In fact, the cargo that he added was moving along the stationary track. That is the microtubules in this experiment. So finally, he wanted to set up this control experiment. So ideally, if cargo moves on the microtubules, in the absence of cargo, there should be no movement seen. Now, why is this control experiment necessary? Because people can always argue that the soluble fraction has something else that is moving, and it is not the cargo that is actually moving. So in order to conclusively prove that it is the cargo that is moving, what he decided to do was he decided to set up a control experiment where he took microtubules, added energy, and just added the soluble fraction that contains all the protein. However, this experiment yielded a very surprising result, wherein he observed that in the test tube under the microscope, in this reaction mixture, the microtubules in itself started moving. Here there are no cargoes, nothing that can move, but this protein fraction along with the ATP somehow resulted in movement of microtubules in itself. So he concluded that there is possible that if you take a glass slide and put in these soluble fractions, which is the all the protein mixture, the microtubules is kind of pushed, and that is how this microtubules results in moving. This protein he identified to be motor proteins, which are basically proteins that can walk along these microtubules or actin like filaments and take cargo along with it from one end to another. So in the same mixture, if he added cargo, then he observed that the motor proteins move on the microtubules, and along with the motor proteins, cargo is also transported. And he identified this motor protein to be these important class of proteins known as kinesins, which since then have been also shown to play a very crucial role in mitosis, which is what we'll see here. So according to this image, we can see that in these interpolar microtubules, what happens is there are kinesins such as kinesin 14 and kinesin 5. Kinesin 14 is a protein that pulls the pole together. So it moves to it attaches to both the poles and pulls it together. However, kinesin 5, what happens is there are two different motor domains. So it is attaching to one protein and moving along the second microtubule. But in kinesin 5, what happens is it's moving across both the microtubules, because of which there is a pushing force. So both these proteins together, kinesin 14 and kinesin 5, because one is pulling and one is pushing, it maintains the integrity of the microtubule spindle network and make sure that it is oriented properly. Now, another important protein is the kinesin 4 or the kinesin 10. Both play similar roles. These are known as chromokinesins because they attach to the sister chromatids. But what they do is instead of moving towards the pole, they move away from the pole. This is what keeps the spindle in the center of the cell, because there is again a pulling force, which we will see later, but there is also a pushing force, which is in the form of kinesin 4 and 10, because of which the cell ensures that the chromosome is perfectly aligned at the center for movement. However, scientists also observed that even in the absence of ATP, the microtubules can move these chromosomes. So there is again some crucial pulling force that we are missing, and they identified this to be two very crucial properties of the microtubule in itself. The first one is that the microtubule is extremely dynamic. This is what I was mentioning previously, because uh, the NDC80 complex attaches to the lateral region and not to the end of the microtubule. This is because there is continuous polymerization and continuous depolymerization. So there is removing of the tubulin units from the end, and then there is addition of the tubulin units as well. This depolymerization at the plus end. So we can recall plus end is towards the chromosome, minus end is towards the spindle pole. Now, towards the plus end, there is extensive depolymerization, and this release of tubulin units also releases GTP, which can be used as the energy that is used for the movement of chromosomes. However, throughout the entire process, NDC80 complex is attached to the microtubule, ensuring proper attachment of the chromosomes as well. There was this experiment that was conducted that conclusively proved that this depolymerization and polymerization creates a force that moves the chromosomes to the poles. So what happened is what they did was they added these speckles, which are tubulin units that are tagged with a fluorescent molecule. Now they added it in very sparse amounts. So only certain regions of the microtubule is tagged, whereas the other regions of the microtubule is not tagged. Here, the ones that are marked in red are the ones that are tagged. And then they plotted this graph, which is plotted as a across in one axis is the time. So it's the same region of the microtubule that they are photographing, but along different time points, as in the lines are what separates these time points from each other. And we can, if you follow one speckle, say we are following this speckle, we can see that as and when the time goes, these speckles keep moving towards the poles. Now, they propose that the reason for this is that if this is the microtubule that is attaching the spindle pole to the chromosomes, here there is removal and here there is addition, and because of which there is a continuous shortening of the microtubules. The other force that they said was acting was the microtubule flux, which is what we discussed, and the other one is microtubule depolymerization, which is wherein here it's continuously depolymerized, because of which there is a pulling force towards the poles as well. So far we have discussed three forces. One is the polar ejection force, that is a push force that pushes the chromosomes away from the spindle pole, and there are two pull forces that moves this chromosome towards the pole. So according to the stage of the mitosis, these are regulated such that it is either equally or equal in both ends, because of which there is a orientation of the chromosomes to the center of the cell. Otherwise, it's a greater pull force, because of which it's pulled to the spindle poles. Now, again, separating the chromosomes, we have previously seen that there are cohesins that are binding the two sister chromatids together. So it is not easy to just pull apart these two sister chromatids one away from each other. So herein also there is a very complicated regulatory network that plays a role. What happens is there is this proteasomal degradation protein known as the APC that is generally inactive in the cell. But in the presence of CDC20, what happens is it becomes activated, and this generally happens in the mitotic phase only. Upon activation of the APC, it is able to ubiquitinate and degrade this other protein, which is a protein known as securin. Securin was initially bound to the separase protein, but now because securin is degraded, separase is free. Separase is actually a protease which is able to selectively go and degrade the cohesin proteins. So in the presence of an active APC, what essentially happens is separase is activated and the separase can go and degrade cohesins, and ultimately this cleaved cohesins helps in the separation of the sister chromatins. But where does this CDC20 suddenly come from in the mitotic phase? What happens is because of an increased transcription of the CDC20, there is increased presence of protein first in the M phase. And again, what happens is MCDK is able to phosphorylate APC, and this enhances its affinity towards CDC20 and it's able to bind it better. And this binding gives it an increased activity. Now, again, as we've seen, there are multiple checkpoints that are present throughout the cell cycle. One such checkpoint that plays a role in mitosis is the spindle assembly checkpoint. This checks whether the spindles are properly associated, if they are properly bound to the chromosomes, and whether now the cell can start separating the sister chromatids apart. One such protein that plays a role in the spindle assembly checkpoint is this protein known as MAD2. Now, if you notice, MAD2 is only associated, the one marked in red over here, is only associated with unattached kinetochores. Otherwise, it is not attached with the chromosomes that are already attached with the spindle pole. Now, if it's attached, it leads to a conformational change, and this MAD2 is now able to diffuse out from the chromosomes and inhibit APC degradation. So cohesin is intact. So unless and until MAD2 binding is lost, the cohesin will not start getting degraded in itself, and because of which a cell cycle will be halted forever until and unless proper orientation happens in the metaphase itself. Now, the final stage of cell cycle mitosis is cytokinesis, which is the division of the cytoplasm equally between the two daughter cells. And this is characterized by a sudden appearance of the cleavage furrow, which has been beautifully imaged in these microscopic depictions shown here. Now, the cleavage furrow arises because of this actin and myosin, two other very important proteins, and they initially form a ring-like structure. This happens as early as anaphase in itself, and this ring is known as the contractile ring, and this accumulates a lot of other protein as well. It's not just made of actin and myosin. There are multiple other proteins that are concentrated at this region. And at the end of anaphase, the ring starts contracting, as in the diameter of the ring gets shorter and shorter, and because of which the cleavage furrow is formed and progresses up until a point wherein the cells are divided into two different daughter cells. So moving on, we'll look at the previous year assignment questions for the week 4 of the course. The first question was the alignment of duplicated chromosomes along the cell equator indicates which of the following stages of mitosis. This is what we've been looking at. There is interphase, and then by late prophase, the nuclear envelope is degraded, because of which the microtubules are now able to properly associate with the chromosomes. And here in the metaphase is where the chromosomes are properly aligned in the center of the cell. So the answer to this question would be metaphase. The second question was, in animal cells, which of the following is a function associated with the centrosomes? Now, we have seen that the centrosomes have two major functions in mitosis. One is that it organizes the spindle pole. It is from where the macrotubules arise. And two, it regulates the cell polarity, because one is the minus end and one is the plus end, and that in itself is creating a bipolar nature. And so centrosomes are very crucial to the regulating of the cell polarity. But the other function that we have not discussed so far has been observed that these centrioles or centrosomes are also capable of organizing and ensuring that uh, appendages like cilia or flagella arises from it. So the other option, regulation of cell motility, which is mediated by appendages like cilia and flagella, are also because of centrosomes, and they play a crucial role. So the answer to this would be all of the above. The next question was, the spindle fiber attaches to the chromosomes at which region? This again, we have seen that there are different regions wherein telomeres are towards the end, the arms of the chromosomes, and the centromeric region is where these proteins accumulate to form the kinetochore, and kinetochore is actually where the microtubules attach. So the answer to this question would be centromere, because centriole is actually centrosomes, telomere is the end, and origin is where DNA replication happens, and centromere is the region where kinetochore assembles. The next question was, the cyclin-dependent kinase of Saccharomyces cerevisiae is called as. This we have seen in the previous class, wherein there are multiple phases and there is only one CDK in yeast, and in this case in budding yeast it is CDC28. Lamin filaments are dissociated by the activity of what? So lamin filaments, like we saw, was the structure that is present to the inner side of the nuclear membrane, and this is degraded by the activity of mCDK. So mCDK is basically this complex of cyclin B and CDK1, which is increasingly active in the M phase only. So the answer to this would be CDK1 and cyclin B, because the other cyclins are present in other regions of the cell cycle. In which phase does the breakdown of nuclear envelope occur? So again, like we saw previously, the nuclear envelope is broken down in the pro-metaphase, which is the stage just after prophase but before metaphase, wherein lamin is extensively phosphorylated and nuclear pore complexes are also disassembled. And this is the stage where the chromosome starts attaching to the spindle pole. So the answer to this would be pro-metaphase. Next question was, which of the following events is not a part of anaphase? So again, if you look at the options and understand anaphase, then it is fairly simple that in anaphase, as the daughter chromosomes are pulled towards the end, the formation of nuclear envelope, however, does not happen during anaphase, happens much later after completely segregating the chromosomes. So if the question is not a part of anaphase, then formation of nuclear envelope. The next question was, the contractile ring during cytokinesis is made up of. Again, this we saw in this session that it is made up of actin, but of course it has other proteins as well. However, tubulin is a part of the microtubules and lamin is a part of the nuclear envelope, and none of these proteins are a part of these contractile rings. Which of the following regarding the mitotic exit network is incorrect? So again, mitotic exit network is something that exists in order to ensure that the cell has completed its mitotic events and now can progress to the next stage of the cell cycle. There are lots of protein that play a major role, but here if you look at the options along with this representation, then CDC14 is bound to NET1 and it's inactive. CDC14 bound to NET1 is indeed inactive. If it's released from NET1, then the CDC14 will be able to ensure mitotic exit. BUB-BFA complex inhibits TEM, and this is also true. BUB-BFA is GAF, which is inhibiting TEM. CDC15 inhibits DF2 and MOB1 complex. This is incorrect because CDC15 is actually activating MOB1 and DBF2 complex. LTE1 is a guanine exchange factor. That is correct. LTE1 is a guanine exchange factor and plays a very like spatial control role in the mitotic exit. So the incorrect statement would be that CDC15 inhibits DTF2 and MOB1 complex. The next question, the final question of the assignment, was that during cytokinesis, what actually happens to the septin collar? Now, the septin collar is present in the yeast, budding yeast, wherein there is initially a collar of protein spawn, and then later this ring is split into two, and here is where actin and myosin proteins will accumulate and form the contractile ring, which will help in cytokinesis. So the answer to this question would be that the septin collar is split into two so that there is actin myosin accumulation between these two rings. Now, moving on, I will briefly discuss what centrosomes are and what their role is in mitosis. Now, we've seen throughout the discussion and in the course content of this week that centrosomes are what forms the spindle pole. Now, each centrosome has this mother and the daughter centriole, and they have multiple proteins present to it and also have this pericentriolar matrix that is again composed of multiple proteins that help in its crucial role. So this is present one per cell. So in early G1 phase, there is only one centriole present in the cell, but then each mother centriole is able to divide and give rise to a daughter centriole each. So in the S phase, wherein the DNA is replicating, in that phase itself, there are two mother centrioles and two daughter centrioles in the cell. Ultimately, in the G2 phase, this division is complete, and when the cell is entering mitosis, as in when mCDK is increasingly being produced, these centrioles and these centrosomes are matured and they move to two different ends of the cell, and this forms the spindle poles. So there are multiple regulatory mechanisms again that is regulating this process, but there have been loss of centrioles that are also seen mainly in plant cells, which is still carrying out mitosis independent of centrioles. And there are also multiple centrioles seen in cells, mainly ciliated cells, such as the cells of the respiratory tract or the reproductive tract, wherein the cells need cilia. And like we've seen, cilia generally arises from centrioles as well. So these cells have multiple centrioles, but otherwise per cell, there is generally only one centriole that is maintained, and only during replication and in the cell cycle, it is giving rise to two. So again, like I mentioned, plant cells do cell division, and they are able to undergo mitosis actually without any centrosomes. So how this happens is first we'll see what are the basic functions of centrosomes. So like we've seen, it first ensures bipolar orientation of the microtubules, as in, like I mentioned, one sister chromatid has to be attached to one end and the other sister chromatid has to be attached to the other end. It also plays a very key role in microtubule nucleation. So like we've seen, microtubule is made up of these, it's basically a polymeric unit. It's made up of these proteins known as tubulin, and it actually nucleates from the centrosome itself, because the centrosome has these proteins known as the gamma Turk complex, which is a bunch of proteins that is able to first nucleate the attachment of multiple tubulin subunits to each other. The other major function that it performs is the minus end stabilization. So minus end of the microtubule is actually attached to the centrosome itself, because of which it is stable, and plus end is relatively depolymerized faster, and the minus end is not depolymerized that fast. Then how do these plant cells undergo mitosis? So the first function, like we saw, was the bipolar orientation. So in order to ensure this, the plant cells have developed other means, wherein first they form a pre-prophase band. Here what happens is there are microtubules that are attached, that's forming like a ring-like complex around the nucleus before the prophase even begins. So that's why it's known as the pre-prophase band. And this they have like the scientists have discovered to be an analog of the centrosome. So from here, there are these bridge microtubules that form, which connect the pre-prophase band to the nuclear envelope. So this helps in orientation of the spindle pole, as in the cell knows where the spindle pole needs to be formed. and also helps in the further generation of the mitotic spindle. So here we can see that what happens is once this preprophase band is formed, there are regions of this that is moving towards the poles. They're known as polar bodies, and these polar bodies then form these mitotic microtubule organizing centers, MTOCs, from which the mitotic spindle arises and attaches to the chromosomes. Later, what happens is once this nucleus is divided into two, there is something known as the phragmoplast, which is formed, which is like a precursor to the formation of the cell wall. So those are also known to be remnants of the MTOCs only. The second function that we saw was nucleation. So like I mentioned, gamma tuck complex is important and it's present in the centrosome, and this is what ensures nucleation and generation of new microtubules. Now, plant cells have known to either recruit these gamma tuck complex to pre-existing entities. So say there is already a preprophase band that is formed, the gamma tuck complex can be recruited to such regions and then nucleate new entities. Or there is also de novo nucleation that has been seen from the plasma membrane. So it's also possible that these gamma tuck like complexes are present near the plasma membrane, and from there these microtubules are generated. The final function of the microtubule was the stabilization of the minus end. And in plant cells, there are many proteins. One such important protein is the SPR2, and these proteins actually associate with the minus end of the microtubules and protects it from any degradation, and in turn gives it its stability. So this is all I had to say in this week. So after this we can discuss, and you can let me know if there's any other things that we'll have to discuss.
Hello, ma'am. Good evening.
Good evening.
Ma'am, I have a doubt regarding cytokinesis. I didn't know whether ma'am had already discussed in the lecture videos. I am a week behind in that. So my doubt was
Okay.
like during like when the division of cytoplasm happens, like how does these different organelles split? Like you have to at one time like for once you have to like split all the like for example mitochondria, everything else other than the nucleus. Like how is there any like or is it random or like you have to give them equally, right? So how does that happen? Is there any process or is it just randomly?
The organelles are also divided. It's not necessarily that it needs to go equally, because the organelles such as mitochondria will also be able to divide by itself. Like there are fission and fusion processes that happens in the mitochondria that by which it can divide on its own. So the cell just needs to ensure that the cytoplasm is equally divided, because of which there is equal distribution of the organelles. I will look into it. That is also regulated. Like you said, it's not random. It's also a very regulated process. I will also look into the course content. If it has not been discussed, then I will discuss it in the next discussion session, like the session that will happen next week.
Okay, ma'am.
So I think we can end this session here if there are no more questions.
Thank you, ma'am. Yes.
Thank you. These videos have been uploaded onto YouTube. I don't know if they gave you the link. If not, I'll put it in the chat box, and you can have a look at it there as well. Have a nice day. Thank you.